Hello, dear students. In last lecture, we have discussed about the nervous system and the neuron. Now, in today's lecture, we will discuss how two neurons communicate, means how the information is being transmitted from one neuron to the other. So let's begin with the introduction to how neurons communicate. As you can see in this diagram, it is showing two different neurons. So we can say the operation of the nervous system, mainly it depends on how well the neurons communicate. Neurons communicate via electrical events that are known as action potentials and also the chemicals which are neurotransmitters. There are many channels across the cell membrane, that is, the boundary between the cells inside and outside. As you can see, cell body is the basically cell membrane is the outer covering of the cell body, which allows positive or negative ions to flow into and out of the cell. Now let's discuss the whole process in more detail, and for that we will discuss the first very term is resting potential. As we have said, there are lot of different ions that have higher concentrations outside the neuron compared to lower concentration inside the neuron or vice versa. As you can see, we are showing in diagram, the extracellular means that is out of the cell region and intracellular signifies which is inside the cell region. So what are the important ions that are present in or out of the neuron? We have cations, anions, and if you see, cations basically include the potassium, sodium and the calcium ions. Anions include chloride and organic ions. If you see the concentration, now we focus on the concentration. The potassium and the organic ions have a higher concentration inside the cell than the outside concentration. We have shown this higher concentration by the bigger size letters and lower concentration by the smaller size letters. So you can see in intracellular region I kept K plus, which is more in size as compared to the K plus of outside the cell. That is mainly just to differentiate between the concentration levels, to show the difference in the concentration levels. Similarly, the concentration of sodium, calcium and chloride ion is much higher outside than the inside neuron. Similarly, we have shown with bigger size letters outside and smaller size letter inside. Now, how we define the resting potential? So, the resting potential is basically related to the concentration differences, or we can say concentration gradients of many ions across the cell membrane. This ionic separation, or we can say difference in concentration of ions at the cell membrane, creates a chemical concentration gradient across the membrane, and the difference in net charge due to the presence of positive and negative ions across the membrane that will create an electrical gradient. And together, the concentration gradient, or we can say chemical gradient, and the electrical gradient, which is due to the presence of charge or difference in net charge, they forms an electrochemical gradient. And this difference in total charge inside and outside the cell, it will create a membrane potential. Now, without any input, the most of the neurons have a stable electric charge difference across their cell membrane. And you can see in this figure, it is showing negative inside and positive outside, because in journal it is more negative inside and more positive outside the cell membrane. This point of membrane potential, where the cell achieves an electrochemical equilibrium, that means at this point the concentration gradient and the electro gradient for each ion is equal and opposite. We'll call it as the resting potential, or it is also termed as the resting membrane potential. Normally, as I mentioned, inside the cell is more negative, and it is approximately 70 millivolt more negative with respect to outside. So we can say that the resting potential or the resting membrane potential is typically minus 70 millivolts. Now, the channels, we have different channels which keep this resting potential. It includes sodium channels, that is inward in direction, and it increase the positive voltage inside the cell. We have potassium channels, and the direction of that is outwards. So it will decrease the positive voltage, mainly the inside cell. It will decrease the positive voltage, means it will make more negative if potassium flows outside. And then we have sodium potassium pump, which will take out the excess sodium which is moved inwards, or it will insert potassium which moved outward, so that it can keep the voltage and concentration of ions steady, means that should be at resting potential without any input. Now we will discuss about the Nernst equation, which is related to each ion. It is the mathematical equation to determine the equilibrium membrane potential, and it is generally represented as Vm for each ion, and it is called as the Nernst equation. Now, for each ion that has single charge, as you can say, we have the sodium ion, potassium ion, chloride ion. They have single charge, whether it is positive or negative. If it is having single charge, then this equilibrium membrane potential can be given as this equation: Vm is equal to plus minus 61.5 into log of the concentration of ion outside the cell to the concentration of ion inside the cell. This is for the ion with a single charge like sodium, potassium and chloride. Similarly, if we have an ion with a double charge, like calcium, we have calcium 2 plus, which is having a double charge, then the membrane potential can be given as again Vm, which is equal to plus minus 30.75. It can be positive, it can be negative, into log of concentration of ion outside the cell to concentration of ion inside the cell. So this is the Nernst equation for the single ion or the double ion charge. Now next we will just brief view will have of ion channels. What are ion channels? They are porous transmembrane proteins which help in regulating the voltage gradient across the cell membrane by allowing ions to diffuse through the pores down their electrochemical gradients. We have two general types of ion channels. They are voltage gated ion channel and the ligand gated ion channel. Now how we can differentiate between the two? So, voltage gated ion channel, transmembrane ion channel that are activated by changes in electrical membrane potential, that is voltage gated channel. And the ligand gated ion channel means the transmembrane protein which opens to allow ions to pass through the membrane in response to the chemical messenger, or we can say some input. We are calling it as a ligand. So we have two different type of ion channels, the voltage gated and the ligand gated ion channels. Now let's discuss about another point, that is action potential. As you can see, the dendrites, which are receiving information, or we can say receiving signals from the other neurons through axon terminals, which release neurotransmitters. You can see that is a synapse. We have discussed that synapse is a junction between the two neurons, or we can say where the electrical or chemical information got interchanged. So in this, we have neurotransmitters. Now, when these neurotransmitters bind to a receptor, you can see in this diagram that neurotransmitters is binding to a receptor in the region what we are calling it as synapse. So when these neurotransmitters bind to the receptors, what happen? The binding open the ion channels that allow charged ions to flow in and out of the cell, converting the chemical signal into an electrical signal. Now, a single neuron is not having a single dendrite. We have tons of dendrites. So, when all the dendrites are receiving signal, and if the combined effect of these multiple dendrites, they change the overall charge, which is enough to cross a threshold, which is typically of minus 55 millivolt, then it triggers an action potential. You can see in this that lot of dendrites, when the signals are combining, signals are coming on the dendrites, then lot of charge will produced, and mainly the positive charge inside, that reduces that resting potential, or I can say it increase the resting potential to more positive side, means from minus 70 to it becomes minus 55, then it triggers an action potential. Now this action potential raises down the axon, triggering the release of neurotransmitters. You can see the action potential is triggering down the axon, and then it will release the neurotransmitters on the other end, and this will further relay the signal. So on this way the two neurons communicate. So we can conclude that neurons use neurotransmitters to signal, or basically neurotransmitters as a signal to communicate with each other, that is cell to cell, and action potential to propagate that signal within the cell. So this is how two neurons communicate with the help of resting potential, various ion channels, and the action potential. Now next we will discuss some terms which are further useful in producing or maintaining the resting potential and the action potential. So in that we have excitatory post-synaptic potential. Now when neurotransmitters bind to the dendrites, we can say, or receptors, they open various ion channels all at once, like sodium ion channel, calcium ion channel, they can flow in. And ions like potassium, they can flow out. Now, if all are added up and the net influx is of positive charge, then we can call it as the excitatory post-synaptic potential. It can be also seen in this way. When a neurotransmitters bind to a receptor on the dendrite, a ligand-gated ion channel, because now it is input, some signal has reached, so that will be a ligand-gated ion channel that opens up. It allows certain ions to flow in. For example, we can say ligand-gated sodium channel allow sodium ions to flow into the cell, and it makes the inside cell less negative because now positive charge is flowing in, so it will make the cell less negative, inside the cell less negative, and it is known as depolarization. Similarly, we have another kind of potential, we can say, that is inhibitory post-synaptic potential. Now in this, the opening of only ligand-gated chloride channel, which makes a net influx of negative charge. Then it is inhibitory post-synaptic potential. And this makes, because it is the flow of negative charge, so it will make inside cell potential more negative, and this process is known as repolarization. Now let's discuss this process in a little bit more detail. This is a graph showing different regions. First is, we can say the cell is at rest or it is resting potential, which is typically minus 70 millivolts. That means in this case, sodium is more outside, potassium ion is more inside, and all the channels are closed. Now what happens? Suddenly some stimulus comes, and if all the stimulus, we can say, they are added up, then they will cross the threshold, which is shown as again minus 55 millivolts typically. Then some of the sodium channels open up. Because of that, sodium comes in, and if threshold is passed, they all will open, and that triggers the action potential. And it will also make the inside cell more positive, and that is depolarization. You can see that 3 basically, you can see that is becoming positive, so it is depolarization. And once it reaches the peak action potential, what will happen? Again, this channels or channel voltage, we can say, it will again start decreasing. In that case, chloride channels will open up. Sodium channels will be closed, potassium channels will be opened, potassium moves out, and the inside cell will become negative, and also chloride channels can be opened up. And this process, which will decrease the potential, we can say, or it will take to negative side, that is repolarization. And what is hyperpolarization? As we have seen that potassium is leaving the cell, and slowly that potassium channels close. Cell becomes more negative than it was at rest. That point is hyperpolarization, means now cell is more negative even with minus 70 millivolt. So that state is hyperpolarization. And to remove this hyperpolarization situation, we are using sodium potassium pumps. And when we are using sodium potassium pump, what we will do is, we will discuss a little bit in more detail in next slide. What we will do, they will take extra sodium outside and they will insert potassium to make the ion concentration steady, or we can say to bring the resting potential again to minus 70 millivolts. So both the sodium and potassium channels will be closed, and then sodium potassium pump will use to return the sodium and potassium to the right places. So this was whole process how it is doing. Now let's discuss a little bit about sodium potassium pump. Now adenosine triphosphate, that is ATP, which is an organic compound or hydrotype that provides energy to drive many processes in living cells. For example, muscle contraction, nerve impulse propagation, condensate dissolution, and chemical synthesis. Now formula for this ATP is C means carbon 10, hydrogen 16, nitrogen 5, oxygen 13, and P3, that is phosphate. So the process of moving this sodium and potassium ions across the cell membrane is an active transport process involving the hydrolysis of ATP, that is adenosine triphosphate, to provide the necessary energy. This is how it happens. That this hydrolysis of ATP, it is converting into ADP plus phosphate, and 3 ions, sodium ions it is producing outside, and 2 potassium it will move inside. It involves an enzyme which is referred to as sodium potassium adenosine phase. This process is responsible for maintaining the large excess of sodium outside the cell and the large excess of potassium ions on the inside cell. It accomplishes the transport of 3 sodium to the outside of the cell, as you can see in the equation, and the transport of 2 potassium ions to the inside, thus in total removing 1 positive charge carrier from the intracellular space. This is the journal, we can say, representation of ATP and how it is getting hydrolyzed. So energy is stored in the phosphoanhydride bonds between the 3 phosphate groups in ATP. This energy is released by ATP hydrolysis, and a water molecule which hydrolyzes this ATP into ADP, that is adenosine diphosphate, and inorganic phosphate by breaking the bond between the 2 terminal phosphate groups in ATP. This reaction also releases energy and a proton. This energy is released because the bond energy in the phosphoanhydride bond is greater than the energy in the bond of products. So you can see by the equation also. So this is all I want to discuss. This is how two neurons communicate and how various channels open and close, and what is the function of sodium potassium pump. So in next lecture we will start with the electrical analog of neuron, how a neuron can be represented as a means of electrical circuit. So for now, thank you so much.